Great. And with that, we're going to start off with session number 1, and I just want to invite Romanos Sklavenitis Pistofidis and Gareth Morgan, who will be co-chairing this section. Gareth actually thankfully stepped in. Mirav Liba couldn't come to the meeting, but Gareth will be substituting. And so, Romanos, Gareth, do you want to come up and... Thank you very much. Yeah. You have the program? Yeah. Is Gareth here? There he is. Gareth. So you guys can turn seats here. And the pointer is weird. You might want to just show when they have to point it down for a second, and then it'll populate.
I'm not sure what's going on here.
So we're just introducing this. Do you want to start?
Yeah, go for it.
All right. Hello, everybody, and welcome to the first session of this meeting. This is about Waldenstrom's predisposition. I am Romanos Sklavenitis Pistofidis. This is Gareth Morgan.
Yep, it's great to be here. Nice to see you all. Great to be on the stage with Romanos here.
Likewise. So the first talk is going to be by Sigurdur Christensen from Landspítali, the National University Hospital of Iceland, and it's going to be on findings from the iStop study.
Thank you so much. Thank you everyone for this, and thanks the organizing committee for inviting me, and of course Chris, who I think is already busy in the VIP room organizing things. I'm here to talk about the iStop MM study. This is actually my second time I'm here in Prague. This is the first time ever I wore a scrub, so I'm here a second-year medical student on a 1-month visit to the University Hospital here in Prague. I might have changed a little bit, so this is me on the right. Super happy. Unfortunately, they placed me on the surgical department, so I was only at the hospital for 1 day. But I spent the rest of the month enjoying Prague, and I love it very much. But now I wasn't invited here to talk about this. I was invited here to talk about the iStop MM study. So the real question here, because I have 10 minutes, I will go right to the chase here. Should we screen for AMCUS is the question we asked ourselves back in 2016. And while we are analyzing and evaluating this in Iceland, we can answer a lot of other questions that are related to AMCUS, and I will now share with you what we have observed for IGM AMCUS. So this is the design of the iStop MM study. We invited all Icelanders 40 years and older at the time to participate, and asked them to give informed consent, and those that would give informed consent would then be analyzed with protein electrophoresis and free light chain analysis, and we actually also did total IgG A&M and total albumin. People with AMCUS would then be randomized to a clinical trial, with arm. It's not a pointer, right? Yeah. So arm 1 is standard of care, if you will. So these individuals are like in the rest of the world. They have AMCUS, but they are not screened and they are not followed. Arm 2 and 3 have different types of intervention strategies, but they are basically informed that they have AMCUS. We meet them and follow them over time. So the key outcomes of the iStop-MM study is really to evaluate the impact of screening on overall survival, but we also do lots of other stuff like try to understand how we should work up these individuals when we meet them and they have MCUS, and how often do we need to follow them, do we need to do bone marrow repeatedly or not, and so on. We also are looking to evaluate and propose new prognostic models for progression. We are doing intensive biobanking, and also we are looking at an early intervention. So the whole project is early detection and early intervention. So this is Iceland. So 148,000 Icelanders were born 1975 and earlier at the time, so 40 years and older. They could give informed consent either by logging on to our homepage, iStopMM.com, or if some people wanted to use a pen and write a paper to us, so they could do that as well. So 80,759 of these people gave us informed consent. So these are the study population. That's it for the participants at the time. They didn't need to do anything else. Just wait, and then the next time they would do a blood test, we would get a serum sample and perform the protein electrophoresis and free light chain analysis, which was actually sent to the binding site in Birmingham. And this is Simon de Rökkvaldsson, who is a postdoc. He is here pretending to draw blood from the participants. But we managed to do 75,422 serum samples. Simon will give you a more detailed presentation of the IgM findings from iStop-MM this Friday during the poster session. So this is what we found. We have 3,725 individuals that were diagnosed with MGUS. It's a bit busy scene here. Thank you, it's fine. So these were randomized into arms 1, 2, and 3, as shown here. And this is the prevalence that we have found for MCUS in our cohort. And as you can see here in, I don't know the color here, but the third from the top is the IgM curve. We can see that approximately 20% of Icelanders have IgM MCUS. Obviously, IgG is more common, and biclonal is, or more than two clones or more is also quite common, as you can see here in purple. So another interesting thing, I think, is when you look at this. So what we did is we did protein electrophoresis, and then we did immunofixation, then free light chain analysis. So we also chose a random cohort of the original iStop-MM, where we did mass spectrometry on these individuals. So we looked at them, and these were just picked randomly. So some of them might have had MCUS, and most of them didn't. And when you look at this, and mass spectrometry is a fascinating method to detect M spikes, but, and this is also a big but, because it's very sensitive, so you pick up a lot. And what you can see here is if you do protein, or if you look for M spikes using mass spectrometry, you will find a lot of M spikes. Most of them will be IgM. So you can see here that I think it's 14% of the population in Iceland has IgM proteins using mass spectrometry. And you can also see that a lot of them, almost, I think it's 11-something%, have more than one spike on mass specs. And when we look at this in more detail, and we look at the people with more than one spike, actually almost all of them had one of the 2 or 3 more spikes were IgM. So IgM M spikes is very common, and if you combine these two, this is what sticks out. So this is nice to present when you're at a Waldenström meeting, but it's difficult when you have this result because it doesn't make any sense. Because, why do a lot of people, like if you look at the very elderly, almost everyone has an IgM M spike using very sensitive methods. So we need to figure out what's going on here. So is this something that is transient? Is this just a normal immunological reaction to an infection, autoimmune disease, or something else? Is this the beginning of IgM AMCUS? So this is below the detection limit of immunofixation, then goes on to develop IgM AMCUS. Also, why would they have so many IgMs? Because IgM is, as I told you, not the most common type. Is there any clinical significance to that? I think that is something that we need to find out, that this is what we're doing now. So we're measuring these same 5,500 individuals 5 years later and re-evaluating if they still have these small peaks, and if it's the same peak, and what is the significance of the peaks. So this is something that is a pivotal point in my talk. This is the animation part of the talk. So I think this is actually, I should give credit again to Simon de Rökkvatsson, who did the animation. So look. Yes. So this is the iStop Waldenström. So these are the results for the iStop WM. So we have 935 individuals that were diagnosed with IgM AMCUS on protein electrophoresis and immunofixation. So this does not include the mass spec. So we have 300 something in each arm with IgM AMCUS. So people with IgM AMCUS in the study, they were randomized, just like any other person with an M spike. We did bone marrow biopsy in every one of them. We did CT of the neck, thorax, and abdomen during follow-up. And if they had LPL more than 10%, so they have Waldenström, then they were followed at least every 4 months for the first year, and then every 6 months after that. And we do annual bone marrow biopsy and aspiration and biobanking these individuals as well. So when we look at these individuals in arm 1, which would represent the standard of care, you don't look for it, you don't follow it. These are on the far left. You can see that you diagnose a little bit of Waldenström and some individuals with non-Hodgkin's lymphomas and CLL. You don't pick up a lot of smoldering Waldenström. And then in arms 2 and 3, the difference between them is we have a lower threshold for repeated bone marrows and more intensive workup strategy. We will find more advanced precursors, more smoldering Waldenström, and more non-Hodgkin lymphoma and CLL. So if you screen, you will find these individuals. This can also be shown here. So if you look at these, the gray one is arm 1, so this is what you are seeing in your home countries, and then you have arms 2 and 3, which is like different shades of purple. So you can see there's a clear difference in more advanced disease, both if you look at smoldering disease and beyond, but even if you just exclude all the smoldering Waldenströms that we diagnose, there still is a trend towards a difference there. So I think this is something that is interesting because we do so many bone marrows. We did bone marrow in everyone in arm 2 and 3. And Icelanders have no problems with turning up to a bone marrow evaluation. So we just send them a letter and tell them that on Wednesday at 10 o'clock, you're scheduled for a bone marrow. And by the way, you have an M spike, and 95% of them will turn up for that bone marrow. So what we're doing now is looking at, should we really do a bone marrow biopsy and aspiration in everyone with IgM AMCUS? Because the current guidelines, which are written by the International Myeloma Working Group, they suggest that everyone with an IgM AMCUS should do a bone marrow evaluation. So only IgG or low-risk AMCUS you can defer from bone marrow. So what we're doing now is looking at this, so you can put in your individual with an M spike, and you give the M spike and the total IgG, A, and M, and the FLC ratio. You can see the probability that this particular individual will have more than 10% plasma cells or LPL in the bone marrow, so this will give you an idea if you should go ahead and do your bone marrow biopsy. Yeah. So this is something that we just, we basically just used to brag. So this is that slide. So I will tell you that if you're interested in looking at IGM-MGUS in any detail, we have a lot of information that we would like to share with people that are interested in doing research, because we don't have the time or resources to do everything ourselves. So this is the kind and the amount of data that we have for these 80,000 individuals, of which some of them have IGM-MGUS. And this is the biobank that we are doing, the ISTOP-WM biobank. So we do a lot of serum sampling and we put it in the freezer. So every time basically we draw blood, bone marrow, or urine, we put something in the freezer for collaborative use. So this is what we call the ISTOP-WM tree, which was very small when we began in 2016. And this keeps on growing, so every time we have a meeting or a discussion, this gives us new ideas and new outcomes to evaluate. And I show this because we really want collaboration with people that have ideas that are interesting. So just send us an email, and this is the ISTOP team, and these are the funders and our collaborators. Thank you very much.
Thanks, Sigurdur. Do you want to take a...
Over there?
Yeah. So, great talk, great way to start a meeting. Can we invite Sonia Bent to talk to us about a large-scale GWAS looking at Waldenstroms and LPL? Hey, Sonia. Welcome. The stage is all yours.
Okay, great. Thank you for this opportunity to speak today, and thank you to the organizers for this opportunity. So family history is a known risk factor for a lot of malignancies, including Waldenstrom's, but the underlying genetic risk factors are not well known. So he set out to look at the genetic variants that were associated with different lymphoid malignancies. We conducted genome-wide association study for lymphoid malignancies as part of the Interlymph Consortium. So as part of the study, we have a little over 49,000 cases of different lymphoid malignancies included in the little over 80,000 controls. Of those, we have about 1,700 Waldenstrom's cases for analysis. So we genotyped our cases and controls with the Luma global screening array, and then imputed to top that. So we have about 15 million variants for analysis. We then replicated our top findings in a smaller, in 4 biobanks, a slightly smaller sample size with about 570 cases. So these are the results from our study. You can see here the y-axis just shows the negative log p-value with associations with Waldenstrom risk, and then the x-axis just shows the chromosomal position of the variants that we examined. So we identified 12 independent SNPs that reached genome-wide significance at 5 × 10⁻⁸, and these were replicated in our 4 biobank studies. So the most significant loci that we discovered were on chromosome 6 at EXOC2/ARF4, and then also on chromosome 14 here. And both of these top loci we had reported previously. So of the 12 variants we discovered, 4 of these are actually very rare variants, but they confer to high risk. So the odds ratios range from like 12 to about 76, as you can see here. But most of the variants we discovered were common variants with relatively low risk, so odds ratios in the range of 1.2 to 1.6. Among the variants we observed, we also saw enrichment for gene regulatory elements in CD19 positive cells, and they also enrichment for immune regulatory pathways. So just to give a couple of examples here, this is just one example. One of the variants that we found to be associated with risk was located on the SP140 gene. So variants in this region have been associated with chronic lymphocytic leukemia, as well as a number of autoimmune diseases. So SP140 is a nuclear protein mainly expressed in leukocytes. It is thought to regulate immune path genes through NF-κB inhibition. So the risk variant that we found to be associated with Waldenström actually leads to skipping of exon 7 in the SP140 and altered splicing of SP140 gene transcripts. So another example, we also found genetic variants associated with Waldenstrom that were found within the MHC or the HLA region of the genome. So we then included the classical HLA alleles as well as the amino acids. And the most significant association we observed was for amino acids at positions 11 and 13 of the HLA DRV1. So these amino acids are located in the epitope binding group of HLA DRV1. These particular amino acids have also been associated with follicular lymphoma as well as several autoimmune diseases such as lupus. So given these findings, you're curious if some of these variants that we found for Waldenstroms are also associated with IGM-MGUS. So we have 150 IGM-MGUS cases in our data set. And so we did a GWAS for IGM-MGUS. And even though our study was relatively small, we did observe that 6 out of the 12 SNPs that we identified for Waldenström were also associated with IGM-MGUS. In fact, as you can see here, the most significant finding was for chromosome 14. So this was a locus that we previously found to be very associated with Waldenström. And you can see here it's also associated with IGM-MGUS with odds ratios greater than 70. So given that we identified several variants associated with Waldenström, we decided to put these variants into a polygenic risk score and then test for the association with Waldenström in independent cohort. So here we use the UK Biobank cohort. As you can see here, when we looked at the PRS, there was a shift in the distribution of the PRS for cases, shown here in sort of bluish greenish color. And you can also see it's pretty unusual. There's a long tail here you can see, and that's due to those rare variants that we identify associated with the risk. So we also just looked to see at the cumulative risk of Waldenström within the UK Biobank by polygenic risk score. And so you can see here that we see actually a large separation risk looking at the top 5% of the PRS versus the middle and 5% of the risk, you can see that the risk is much higher if you're in the top 5% of that PRS. So Waldenstrom's, as everyone knows, is a rare disease, and so the absolute risk is not terribly large. It's actually quite small. Everyone can imagine that it may have potential utility in more of a high-risk population where the prevalence is higher. So finally, I'd just like to thank the many people that were involved in this project. This is not a complete list, but I'd like to thank in particular the study PIs who were very gracious in allowing us to use their cases for this study, and also Mary Lou McMaster, who isn't here today, but and also Hanle Park, who led a lot of this work. Thank you.
Great talk, by the way. We'll take questions at the end. Do you want to join us again?
Thank you very much for the wonderful presentation. So the next talk is going to be by Stephen Ansel from the Mayo Clinic on genome-wide DNA methylation analysis that reveals epigenetic influence on intracellular and cytokine signaling pathways in Waldenstrom's compared to IgM gas. Welcome.
Well, good morning, everybody. Thank you very much for being here. Thank you, too, to the organizers for inviting me. And I have the opportunity to talk about genome-wide DNA methylation. This is in Waldenstrom's compared to IgM-MGUS, and I am specifically going to highlight some of the work we do showing epigenetic influence on intracellular signaling and cytokine pathways. So firstly, I just need to disclose that my institution does get money from these companies for clinical trials, none of which is relevant to this particular talk. So just reminding you about epigenetic regulation in Waldenstrom's, but kind of more generally, you can see here that if really two components that regulate this, methylation and then histone deacetylation, but methylation really is related to causing nucleosomes to pack tightly together. That avoids the ability for there to be appropriate transcription, and so genes are really not expressed. In contrast, histone acetylation results in far looser packing, therefore more exposure of the genome, allowing for better transcription as the DNA can be accessed and genes can be expressed. Now in cancer, that actually is quite obviously abnormal, and you can see that you can have both hypermethylation and hypomethylation of the DNA. If you have hypomethylation, there's actually a potential increase in transcription as more of the genes are accessible, and there's actually a promotion of tumor growth and survival. In contrast, if there is hypermethylation, genome is not really accessible. It actually prevents transcription, and particularly tumor suppressor genes are therefore not accessible and may not be able to prevent malignancies. So we were interested to see genome-wide whether DNA methylation was a significant issue, but always important to look back at the literature to see what's out there. And originally in Waldenstrom's, there's an assessment of the promoter methylation, specifically of genes that regulate CXCR4 and MYD88, and this was work done by Zach Hunter. And you can see that across Waldenstrom's, there is a differential DNA methylation that was already shown now almost 10 years ago. A more kind of comprehensive analysis was done where Dr. Roos Weil actually did, and their group did DNA methylation and showed two different subtypes in Waldenstrom's. One with more of a plasmacytic differentiated phenotype, and one with more of a memory B-cell phenotype. As you can see here, those that were more memory B-cell-like associated more with CXCR4 and MYD88 mutations. Those with PC or plasma cell-like findings more associated with 6q deletions and increased expression of CD38. So we were interested to see across the entire spectrum of various IgM immunoglobulins and gammopathies, whether there was a difference, focusing now on DNA methylation. And you can see specifically we were interested to determine whether if we looked at DNA methylation but coupled that with the transcriptome and genome analysis, could we see whether DNA methylation was associated with increased genomic accessibility and increased transcriptomic activity. So in essence, what we did in the study was to compare normal controls to any of the IgM gammopathies, and then compared IgM MGUS to Waldenstrom's macroglobulinemia, specifically looking at DNA methylation. And our goal here was really to look not just at the DNA methylation patterns, but also the effect on the genome and transcriptome. So first of all here, this is an assessment of all of the IgM gammopathies, and as I might have said, well, I didn't say in the previous slide, 23 Waldenstrom's patients, 6 IgM MGUS, and 5 controls. So here's the analysis of IgM gammopathy compared to control, and you can see that there is a significant difference, particularly in differentiated methylated regions, and specifically there's a greater number of hypomethylated DMRs in the 5' UTR, the intron and CPG shore regions, and conversely there's a greater hypermethylation seen in promoter 5' UTR, exon and CPG regions. But more interestingly is how does this really apply to the genome and transcriptomic activity, and you can see there's actually activation of multiple members of the interleukin families that we can see when we couple this with the RNA-seq analysis. You can also see there's upregulation of JAK-STAT pathways and the PI3 kinase pathway, and downregulation of P10, and enrichment for CXCR4 signaling. We then did the comparison of Waldenstrom's to IgM MGUS and showed that, interestingly, it was almost the opposite picture, where there was a greater number of hypomethylated DMRs in the 3' UTR, the intron, exon, and CPG shore regions, and a greater number of hypermethylated DMRs in the 5' and CPG island regions. And this translated into actually an overall inactivation of cytokine and immune regulation pathways of IL-6, IL-8, and IL-15, and inactivation of particularly STAT3 signaling, and promoter hypermethylation of EGFR, FGFR2, FLT4, and others. So I wanted to put that in context because this was a little bit sort of unexpected, but you're going to be hearing from my colleague, Dr. Mondello, in just a few presentations that last year we had presented and published data about 3 different clusters that we can see in Waldenstrom's. This was utilizing a multi-omics approach where we looked at not only the malignant B cells and plasma cells, but also matched bone marrow supernatants and serum specimens, and did a multi-omic analysis. And you can see the principal components analysis on the right, these clusters separately, but specifically that you can see that C1 is predominantly Waldenstrom's, C2 is a kind of a mixture of Waldenstrom's and MGUS, and C3 looks more kind of normal phenotype-like. And we'd shown that C2 was actually very much cytokine-driven. So then we took this DNA methylation analysis that I just showed you and connected that with an association for these clusters. And here's what we found here, but at the bottom line is that's important is to note that C2 was noted with the upregulation of inflammatory response proteins, very consistent with what we published before. And in fact, there was an opposite trend in C1 with decreased inflammation and more increased proliferation. So what does this all mean? Well, I thought that the editorial that was written to go with the paper I'm just presenting was very informative. This really just tried to take all of this work that I just showed you and put it in context. Importantly, what you can see across the top is as you go from healthy to IgM MGUS to Waldenstrom's, as the increased number of plasma cells and the migration from memory-like B cell to plasma cell type phenotype develops, this is associated with the increased expression of the MYD88 and CXCR4, as you can see here, and finally, as you get to this more mature Waldenstrom's picture, you start to get more of the deletion 6q. As you can tell, these 3 clusters we spoke about Cluster 1, which is more Waldenstrom's-like, is predominantly with this particular phenotype. Cluster 2 with more cytokines, kind of in the intermediate MGUS to Waldenstrom's, and cluster 3 more kind of a phenotype similar to normal. And here you can see how that then fits with the DNA methylation. You can see the methylation is predominantly here in clusters 3 and 2 and going down and becoming less as it becomes Waldenstrom's as the proliferation and growth of the tumor increases. And this inflammatory picture that we can see fits more in this cluster 2 IgM-MGUS to Waldenstrom's transition. So what do we conclude from all of this? Well, clearly there are differential methylation patterns seen in IgM-MGUS compared to Waldenstrom's. Multiple cellular pathways, particularly cytokine pathways, upregulated and clearly epigenetically regulated. This clearly is part of the progression pattern that we see and provides an opportunity because there are multiple therapies that could potentially be brought to bear. So with that, I'll end and thank the collaborators working along with me, particularly Dr. Chohan did a lot of the work, Dr. Paludo, Dr. Mandela, Dr. Abikun are all at this meeting, and thank them for their contributions. Thank the Waldenstrom's Foundation for the funding, and you for your time and attention.
Thank you so much. Continuing a great session, I think. I think it's the last one of the morning, and this time Simone Ferrero on multiomics for deciphering mechanisms.
Thank you.
Look forward to a great talk.
Thank you all, and good morning, and thank you for the kind invitation. This is not a simple topic, because as we know, the biological mechanism driving the progression of the premalignant IgM-MGUS clone to Waldenstrom are still unknown. What we know is that the risk of progression from IgM-MGUS to cancer is about 2% per year, and overall the 20 years risk of progression is about from 20 to 50%, depending on patients' risk factors. But which are the risk factors? We already know a lot about the Waldenstrom genomic profile, defined mainly by MYD88, 604, TP53 mutations, but this mutation does not add relevant insights regarding the issue of progression from asymptomatic to symptomatic Waldenstrom. So how a multiomics approach might investigate the transition from IgM-MGUS to Waldenstrom? Actually, in the last years, several reports have been published and will be discussed today about mutational analysis, about gene expression analysis. Professor Ansel described as the DNA methylation profile, and there are also many reports about microenvironmental alterations in the progression from MGUS to Waldenstrom. And in this context, there is also an important challenge of proceeding to a non-invasive diagnostic approach to understand these issues, mainly based on peripheral blood samples and not bone marrow, and the analysis of cell-free DNA and cell-free RNA. So what about genomics? We already know a lot about mutational and cytogenetic landscape of Waldenstrom, but it is particularly interesting report that has been published last year, focusing on a comprehensive multiomics analysis of quite a relative small number of MGUS and Waldenstrom patients. But the authors, they were able to identify 3 molecular clusters with distinct multiomics characteristics. From a genetic, from a mutational point of view, you can see different mutations in the cluster 1, the red one, 2, and 3. But in particular, these 3 different clusters, as already said, have a differential gene expression profile. And to simplify, the cluster 1 is mainly represented by Waldenstrom patients. The cluster 3 is mainly represented by healthy subjects, but also there are some Waldenstrom patients inside, and the cluster 2 in the middle is mainly represented by IgM-MGUS, but also some Waldenstrom patients. So some light has been shed in this issue, but there is also something to be better clarified. DNA methylation, as already said, made help in having a better landscape. This report about 23 patients with Waldenstrom, 6 with IgM gas, and 5 healthy patients, investigated RNA sequencing, whole exome sequencing, and specifically DNA methylation. And the authors were able, as said, to identify differences in methylation patterns of IgM gammopathies versus controls, and in particular of Waldenstrom, that is hypermethylated versus IgM gas. Moreover, several pathways linked to the immunoregulation have been described and were differentially regulated on the basis of these methylation levels. Finally, there are many reports, there are at least 2 reports from different groups about alterations in immune microenvironment in Waldenström macroglobulinemia. And in particular, I mean, there is a differential landscape of non-cancer cells. I mean, T cells, B cells, NK, and also myeloid cells in Waldenstrom, but they are already alterated in M-gas status, and they are, of course, not alterated in healthy patients. So also alterations in non-neoplastic cells, in microenvironmental cells, have been associated with the progression from M-gas to Waldenstrom. And in this context, this is particularly interesting, the data recently published on CHIP, clonal hematopoiesis of indeterminate potential. This CHIP was studied in a large series of asymptomatic Waldenstrom. The frequency of clonal hematopoiesis is about 13%, both in M-gas and in smoldering Waldenstrom. The classical genes of the CHIP were described, like DNMT3A, TET2, and ASXL1. And what is particularly interesting is that patients carrying these CHIP mutations have a higher risk of progression from asymptomatic to symptomatic Waldenstrom. And this is particularly relevant. But why I'm discussing with you of these issues? Because in Italy, in Spain, thanks to the Italian Lymphoma Foundation and a grant from IWMF, we started collecting samples from IgM gas and Waldenstrom patients some years ago. We, in the field bio-WVM trial, we collected 300 patients. We already presented the preliminary results of non-invasive diagnostics in some meetings, and we are going also to present some updates in this meeting. But 2 years ago, we applied to the Robert Kyle Development Award to use these samples, the leftover of these samples, also to analyze the progression mechanism from M-gas to Waldenstrom. And briefly, this is the plan of our projects. We have 70 patients left over from 70 M-gas patients, from 160 asymptomatic Waldenstrom and 70 symptomatic Waldenstrom. We have bone marrow, peripheral blood, and plasma samples from these patients. And we followed up these patients at least for 5 years with sequential sampling. We have three aims. First of all, to investigate novel specific genetic drivers of progression, then non-invasive cell-free RNA biomarkers, and finally CHIP. Regarding the task 1, we are going to analyze by droplet digital PCR in already collected unsorted bone marrow leftovers the mutations, the alterations of 2 genes that were not covered by the initial planned NGS panel, that is PRM1 and TNF-AP3. These are the preliminary results. We investigated around 90 bone marrow samples, and we were able to identify about 12% of copy number variation of these genes that might be involved in the progression mechanism. The task 2, regarding non-invasive cell-free RNA, We know that we have currently no data on circulating coding and non-coding RNA, and we are going to investigate these because circulating RNA products might be involved in the key pathways of lymphoma initiation, progression, and also transformations. Overall, we investigated 15 patients, plasma samples of 15 patients of IgM gas, 32 patients of asymptomatic Waldenstrom, and 13 patients of symptomatic Waldenstrom, along with 28 healthy controls. The goal was to compare these different categories and to follow up the alterations of gene expression in different follow-up. This was made in collaboration with the University of Ghent. Please see the e-poster tomorrow of Philippe Ducrener. He will explain you the first results of our study. In particular, we were able to identify higher cell-free RNA concentrations in Waldenstrom, in symptomatic Waldenstrom, than asymptomatic, than MGAS. And we were able to identify that expression of peculiar genes, as for example, this one, PF4V1, might be associated, maybe a biomarker to identify Waldenstrom versus MGAS or controls. And finally, we're going to investigate a chip in a large proportion of patients. We have samples at baseline, after 1 year, 2 years, and we are currently collecting samples at 5 years follow-up because, as I already said, there are chip is associated to an increased risk of progression, but we need more data on prospective sample series, prospective patient series to better understand this issue. And these are the acknowledgements of all the people involved in these studies, the University of Torino, of Pavia, Salamanca, and Ghent, the Italian Lymphoma Foundation, and the IWMF for the funding. Thank you very much.
So a great session. We'd like to open it up for questions from the audience. Perhaps me and Romanus could start off. It was a session on predisposition to Waldenstroms and progression from Waldenstroms. Perhaps one of you'd like to address the question of how would you see that being used clinically, and how is that going to improve the management of patients with Waldenstroms? Perhaps I'll ask Sigurd, since I was with him last night.
So what's the question?
What's the point?
So I think it's very important to distinguish like the point of basic research, trying to understand what is happening. Why do people develop MGUS, IgM MGUS, and who of all these individuals with IgM MGUS go on and develop Waldenstrom? So I think a lot of these, like you presented about the IRF4, is something that makes sense. So the German variant in IRF4, it has to do with long-lived plasma cells and germinal center building, so that makes sense. So this germinal variant increases your risk of getting IgM MGUS and Waldenstrom. I mean, I think still it helps us understand the disease, but it's not helpful in the clinic. It's not something that we use for, clinical counseling. It's not something that we need to use to understand, like, who should do repeated follow-up and who can be sort of omitted from follow-up. And so a lot of these basic things that are going on, time will tell which of them will help us in the clinic, but they're definitely super important for us to understand, like, the targets and how, what to go on and develop targeted therapy and so on.
Steve?
I would totally agree with that point, but I would also say that we're actually, as we understand those targets and can really work out, so for example, using DNA methylation as an example, multiple epigenetic modifiers that are clinically available. So could it be that we could identify specific patients where they are clearly on a track to develop Waldenstroms and intervene by actually taking patients with MGUS and keeping them MGUS rather than have them become Waldenstroms. I certainly think there's potential merit there. I think the important point, as my colleague just said, is that we really need to understand who are the people at real risk, because we don't want to put everybody on medications they don't need.
So just before we go to the audience, or Romanos, I'd like to ask Sonia to comment on SP140 and what she thinks of the relevance of that, because we found it in a mutation analysis, so I was fascinated.
Sure. Yeah, for Waldenström, it's relatively low. That's actually it's not great. But it's interesting, it's a functional variant. It's the same variant that you see with CLL. So I think it's a really interesting gene, and we'll also look at it in more detail.
Thank you. I have a quick question for Stephen, actually, in terms of the usefulness of methylation data. Did you ever see in your data that there's a signal that is not captured by your RNA data? Do you foresee methylation data actually making its way to the clinic as a stratification tool?
Yeah, so I think, again, a good question. I think in general, we're really trying to do a multi-omics approach, just exactly as you're pointing out that sometimes things that appear relevant, let's say on a methylation screen, aren't really that relevant when you actually look at what the transcriptome and other things look like. So I think as we move forward, the more we can actually do a multiple analysis, as it were, identify things that appear genomically to be important, but actually transcriptomically result in something valuable, and subsequently, to be frank, proteomics and metabolomics really confirming that lets us really have a good assessment of who and what we need to really target. I think sometimes you can get patterns, and patterns are interesting but not necessarily valuable. So I think in our data, we're becoming more and more convinced that this inflammatory environment is a transitional step, and I think that's where the interest may lie.
Thank you.
So to the audience before the queue gets too long.
All right, Remo Birre, Indianapolis. Thanks for such a great presentation. So my question, I have two questions, but sort of the same question. I'm trying to put all of these talks together in my head and come up with some sort of hypothesis of how Waldenström develops from maybe the patient who has MGUS to the patient who develops the disease. Taking all of this data that you are presented here, what would be a reasonable hypothesis that I could take home with as of today? I understand the work is still going on, but how can I put this all together? And the second part of my question is, a lot of the focus has been on the patient who has IgM MGUS, potentially developing Waldenström's. But what causes the IgM MGUS? What are the hypotheses? And how do we start from before the patient has IgM MGUS? So I'm hoping that all these professors will give me a good hypothesis. Thank you.
I'll start, but hypotheses are just that, hypotheses to be proved. But I think some of the data and what we're thinking about is that as we move from a healthy normal B cell to an IgM MGUS, there is a multifaceted, microenvironmentally driven inflammatory process that's going on. And that increasing inflammation, whether it be entirely from DNA methylation or whether it be a reactive process from the microenvironment, results in a process where there's increased growth and proliferation and development of a more plasmacytic kind of process, making more IgM over time. And then as you move to Waldenström's, you actually lose this microenvironment and gain more autonomy as the cells now become more proliferative, and actually a lot of that cytokine activation decreases. So there's a kind of a peaking in the inflammatory environment and then a decrease as the cells become increasingly autonomous. But that's just a hypothesis with a few clues that doesn't necessarily have all the information. I'll let others who are more expert, as expert.
I'm afraid that this is the question that we all are trying to answer here. Okay, I have a question about multiclonal, the presence of, I assume that these multiple peaks that we have here, because we can have several different peaks with the same clone, just based on alternative splicing. And obviously, this can be done with patients that have IgM monoclonal component, then they can have other monoclonal components. This is just a precision about this topic. And my question is for Sonia, is about the SNPs that they have found in the 14q3213 place. My question is if it is related with the several specific VH segments that are more frequently seen in Waldenstrom's macroglobulinemia, for instance, VH323 or the 330. Are they related with this specific VH segments?
That's a good question. We haven't looked at that yet, so I don't know. It's a good question. We should look at that.
Okay.
Thank you. Bruno Bayo from Pamplona, Spain. I have two questions, if possible.
Go for it.
The first is to Sigurdur. Do you have any insight from your iStop-MM cohort if the rate of transformation in IgM-MGUS is higher as compared to the non-IgM?
No, actually it's very similar to the other.
So different from our previous understanding?
Yeah, it's
Okay.
very similar.
Okay. And the second is to Stephen. If I understood correctly, in cluster 1 you have a predominance of a plasma cell phenotype as the diseases progress. But it is also my understanding that, at least from the cellular point of view in the marrow, when there is a transformation into active Waldenströms, there is an expansion mainly of B cells, perhaps much more than plasma cells. So would this be a B cell with an epigenetic and transcriptional plasma cell phenotype, or truly plasma cells that expand?
Yeah, good question. And actually, Dr. Mondello, I think, will be addressing that in the next session to a greater degree. But I think your point is it's not a pure plasma cell, it's a plasmacytoid cell, if you like, with B cells with more transcriptional elements that are plasmacytic in nature.
Thank you.
Hi, my question is towards Sonia. So I was wondering whether you have had the chance to functionally characterize the germline alteration that you have seen associated with, Waldenstrom, and whether you can speculate, assuming that there is alteration in some of these genes, might be a reason to start a treatment up front to avoid transformation toward the final disease.
So we've looked at the functional mechanisms of some of them. I would say some of them we don't really know the mechanism. I think chromosome 14 is a good example. It's not really clear what the functional mechanism behind that was, whereas other ones is a clear signal. The one at SP140, for example. There's also one near PEX5, which also the functional mechanism is clear, but that was, so it varies. I think some of the variants, there's a clear functional story, and then the other ones, we're not really sure how the variant is altering risk.
Okay, thank you.
You have time for one more?
Yeah, go for it, Zach, we're good.
So this is a question for Christensen, and I was really fascinated by the prevalence of IgM, right? Waldenstrom's is not that common, but IgM-MGUS apparently is everywhere. And one of the things I was curious about is, do you have any evidence that MGUS is really one entity? Or is this more of like a basket where, in fact, some of these IgM-MGUSs, are never going to become Waldenstrom's, and some of them, are much higher risk. Do you have any evidence of that in your data, or do you have any comment just on the general prevalence of IgM?
No, and also it's dependent on what method you use. I think Irene Gabriel has also already published a lot of this in her paper on using mass spec in her cohort. She also finds a lot of IgM-MGUS, and we still need to figure out if they have anything to do with MGUS. Is it just that we all have a lot of IgM spikes, so this is like polyclonals is just a lot of monoclonals? So is it just immune system working, and then we sort of think of this as a monoclonal spike, and because we're all interested in IgM-MGUS, but the real IgM-MGUS that we identify using immunofixation we still need to figure out like the significance of those, just like we need to figure out the significance of AMCUS in general. So, I mean, yes, it is a part of the whole spectrum, but it's biologically different, as we are all aware. That's why we have this meeting here, and we're not only part of the IMS. So this is something that, you know, we together need to figure out.
Yeah, we'll take this as a
One
last
more?
question.
Okay, great. This question is for Stephen. So just really love how you've incorporated the cluster analysis multi-omic with the methylation. And I think it was that last summary slide that you showed that the cluster, seem to have ordered the clusters temporally. And I'm just wondering, for some clarification, is that the case, or is it that some of these are more stable, that aren't really progressing to the more plasmacytic phenotype? Do we know this? Because it is quite fascinating then that this sort of intermediate phase would be driven by an inflammation phenotype, which then seems to resolve at a later phase. So I just wondering if you can comment on that.
That's a great question, and I will say we ordered it that way because it seems convenient and it fits very nicely with a discussion. But your point about is it more of an unstable phenotype versus a more stable phenotype is very relevant. The only reason that we put the inflammatory, shall we say, along and called it a transitional step is that most of the IgM gusses fit there. So the thinking is, well, that seems to be a transitional step, so hence it is. But could it be the opposite, where you're just saying that there's actually just two roads? One is more of a plasmacytic stable phenotype, the other is more of a memory B cell, shall we say, but more transitional or inflammatory stable state, but unstable? Don't know, but I think it's a great question.
Thanks.
So a terrific session. We're bang on time, and so it's my job to invite Bruno and Chris Oakes up to the stage to do the second session. Thank you, guys.
Thank you.